Myself, Venu Vangala, a lecturer at the Center for Pharmaceutical Engineering Science, Bradford. I take this opportunity to convey a warm welcome to today's CPSE seminar at Bradford. I can see many friends, research collaborators, academics, and also some industry friends join today, including Bradford community and students. I welcome you all to this seminar. It gives me an immense pleasure to welcome our very own Professor Gautam Desiraju, who is the leader and the visionary scientist in crystal engineering and who has kindly accepted to deliver a talk to us at the Bradford shortly. It's a great honor to the CPS and Bradford for this talk, and personally, it is always been. I'm always grateful to this Raju as my PhD advisor, and today it is no different. Thank you, sir, for accepting this invitation. So. Today, Professor Anand Paradkar is chairing this session, who is leading the Center for Pharmaceutical Engineering Science since its inception. So before I hand over the microphone to Prof. Anand, a couple of housekeeping items. So please keep yourself in muted and also switch off the video just in case if there are any bandwidth problems. And I checked with Professor Desiraju that is it fine to have some questions. He said at the end of the session, he's happy to take up the questions. On this note, without further ado, Professor Anand.
Okay, thank you, Venu. It is as a director for Center for Pharmaceutical Engineering Science on behalf of University of Bradford. I welcome Professor Desiraju. Thank you very much for giving us this opportunity, and thank you for all the delegates who have joined all across the world. Very eminent August gathering we have. I won't make any I don't think I'm able to introduce Professor Desiraju. He doesn't need any introduction. The world leader in crystal engineering we have for our CPS seminar. That is great honor for us. Please, session is yours, sir.
Right. Let me see. Try the share content now.
Yeah.
Do you see this?
Yes, it is loading.
Yeah.
Now we can. Yeah.
You see this now?
Yes.
You can see the mouse and so on.
Yes, everything. Yes, sir.
Okay, and how about the audio, video?
Yes, everything is
perfect, sir.
Right. Okay. Well, here we go. Professor Paradkar, Dr. Vangala, I would like to thank you for this invitation, and I'm speaking to you in strange times, and good to see so many friends across the continents. I see at least one person from North America and one from South America. So I'd like to thank you especially for this waking up at this early hour and logging in here. And in a sense, yes, I think more people can get together at times like this, thanks to internet and all these technical things. We've all got used to speaking in this format now. Now, for about 50 minutes or so, I'd like to say a little. I know I get an echo somewhere. I mean, is it from my side?
Yeah, could you please unmute if anybody has not done yet? Thank you.
Okay, and so I'd like to basically trace this whole business of pharmaceuticals, especially as it concerns solid forms and crystal engineering. Do you hear me, Venu?
Yes, sir.
Yes, sir.
It is clear. Yeah,
it is clear.
Yeah.
And I'd like to trace a little bit of this whole story that has been going on, not really in a review fashion, because too much has been written and said in the last 20 years or so. But coming to the important thing first, the main problem is that drug discovery and development is both long and risky. And especially with all the time taken in the phase 3 stages and the very low probability of getting an FDA approved drug, which then goes to the market, the whole enterprise has become very, very costly. And this is true in all the countries, including very rich countries. To really look at the amount of time also spent, time is money. 72% of the time spent in the clinical trials, and here there is a logarithmic scale. Roughly the number of new molecules approved per 1 billion global R&D spending. This is corrected dollars, so that the scales for the money are the same. You can really see the new approvals are falling. So it's taking more and more money to get a new drug from scratch. So R&D funds are not available. In general, yes. After COVID, even more so. New drug approvals declining, risk factors. So all these things have led to, this is a real crisis. I think this word is not understated here. And we then come to the idea of crystal engineering.
कोई बॉडी नहीं एकदम टाइम से बिठा दे हम उधर रेस्ट करना है चल बेटा
Hello, Venu, can you mute everybody from your side?
Yes, I am trying to mute that. Sorry about that.
Because it's quite distracting. So really what is, I think most of the people in the audience do not need this slide. But I would like to draw your attention to these diverse areas. It is this part of crystal engineering that has become more and more predominant in recent times. So, and if you go to crystal engineering, the heart of the design strategy are really the supramolecular synthons. This is again review material. And once again, I would feel that most of the people in the audience know about this, so I'm not going to really spend too much, but they are the key elements in the design strategy, especially when it concerns cocrystals, which is the topic of my lecture today. And so then the landmark paper is really the Almarson-Zavorotko review in ChemCom in 2004. And they went from, the synthon of 1995 to the heterosynthon of 2004. Of course, heterosynthons were already known. I was telling someone recently that, Schmidt and Lazarovitz already talked about the acid amide synthon in the early 1970s. It was not called heterosynthon, obviously, and it wasn't called anything, but it was there all the time, and they realized the importance of this, what we call today the acid amide synthon, which you see over here in Zavorotko's paper. So the idea that it is, you have to identify a heterosynthon to be able to design a co-crystal, I think this is the key thing that this particular review in ChemCom really represented. So the whole thing really took off from this point. Now, so if you see this particular thing, these were some of the important papers that followed quickly thereafter, which started citing this paper. And as you can see, the citations for some of the other papers are almost as impressive as the citation is original. So this is clearly, this review was very timely, because you can see the way things took off after this particular thing. And so there was a lot of activity on cocrystals and what the implications for the pharmaceutical industry. Now, in 2011, the FDA came up with a guideline which many of us in the field recognized as being retrograde. And they said something about crystalline materials composed of 2 or more molecules in the same crystal lattice. I mean, even purists will argue about the use of the word lattice itself, and that's another matter altogether. So after this, there was a considerable amount of activity, I think basically protesting these guidelines. And there was a development in 2016, and finally in 18, they came up with a much more scientifically, I would say, reasonable notification, where they acknowledge that the pharmaceutical co-crystals have opened up opportunities for engineering. See this word engineering. Solid state forms beyond conventional of an active API such as salts and polymorphs. So they are like salts and polymorphs, but they go beyond. That was the important thing about this 2018 regulatory classification. So, and I think people then forgot very quickly, but just as an interesting sidelight, what happened between 2011 and 2018 to make them change their stance? This was, I think, a rather important meeting held in the outskirts of Delhi in 2011, shortly after this first FDA guideline came, and it was part of an Indo-USA collaborative program. All the people who attended here are from India or from the USA. And the whole meeting, 2.5 to 3 days, only discussed this FDA definition, nothing else. People didn't talk about their research and this, that, not the usual type of conference. Everybody talked about the FDA guideline and how bad it was. So, and I think some of the people, including Venu, for example, here, many people who are in this audience were present at this meeting. And I think probably in crystal growth and design, maybe this holds the record for the largest number of authors in a given paper. And incidentally, everyone I think who is an author here was actually present at this meeting in a small place called Manesar, just outside Delhi. So a definition was proposed, and I'm happy to say that the 2018 regulation largely takes up the spirit of this definition that was given at that meeting. And so that you can see that because of this 2018 thing, the number of papers being published in the area of pharmaceutical co-crystals is really increasing today. I mean, look at this. I mean, this is just 2012, which is about 10 years ago. And then you can see this is really growing now. It's really growing after 2018. And just for fun, I went to SciFinder last week, and I just pulled out 75 papers which have the words pharmaceutical co-crystals either in the title or the topic, and just to find out on what these papers were trying to describe. You can just look at the wide variety of things. I mean, starting from 2-chloro-4-nitrobenzoic acid, you go to spray drying, tablet excipients, theophylline, hot melt extrusion. What is TAK020? Even I don't know. Continuous manufacture, this is important. In silico screening, syringic acid, ambrisentan. I mean, look at this diversity. You know, the diversity word came in that crystal engineering first slide. And these very, very diverse things seem to be happening. What is happening here? This is not just your crystal structures, synthons, weak hydrogen bonding. No, it's not that. It is, its subject is actually moving quite fast and moving in a very exciting direction. Now, actually, this is not so recent. I think there is a very interesting, and I would feel in hindsight, important paper way back in 2007 by this gentleman called Chow in There's another chow here, Hong Kong, Canada, Macau. And you see, they bring up the idea here that crystal engineering, as defined by so many people before, it provides a rational approach to solving formulation. Now look at this, formulation, processing, product performance. So this paper takes this crystal engineering approach from chemistry to chemical engineering. But once again, if you read this paper carefully, you will find that at the root of his analysis are those same intermolecular interactions again. So he says, how can I, for example, use an intermolecular interaction to define particle size, to define this, this melt extrusion and things like that. So it is a micro level analysis of a macro level issue, which is very, very important industrially. So he has a very nice picture here, a flow chart, which he says relationship between crystal and process design in new drug development. Look at the date, 2008, 12, 13 years ago. Now, this yellow thing is what we always like to follow. Active molecule, salt selection, polymorph. Yes, they were willing to give some concession to polymorph after ritonavir and Zantac. Okay, then API characterization, then API synthesis, scale up, process control, clinical, process, finished product. So this looks fairly conventional, and this is what was going on all the time. But then he has put something called future step. And please notice that the black arrows means succeed, proceed. The dashed blue arrows means failure, go back. So he also recognizes that in this salt selection and polymorph, you may have to go back. So look at the blue arrows here. And in all these places, he has been wise enough to put the blue arrows everywhere. So these things may not go right. That's what he's saying. Now look at what he has put below. It was 2004 when Zeverotko published the ChemCom review. Already in 4 years, he has put cocrystals here. And he's got amorphous also and co-process, which is very important. So look at this, co-crystals goes back in a blue arrow to active molecule. What does this mean? It means that he is feeling already in 2008 that these co-crystals, choice of them, their identity may define, again, redefine what the active molecule is. Of course, here he has co-crystals going in a black arrow to polymorph, which means this is the successful part. But I want you to look at the blue arrows more than anything else. The same story. This is the chemistry side. This is the chemical engineering side. Now look at these blue arrows again. Again, functional excipient, size reduction, nanoparticle, amorphous. So these things are also going to be important. And now I hope you can have a feeling and appreciate the diverse nature of those recent papers that I pulled out from SciFinder. So there are lots of people working not only on this, but on this and on this and so on. So this whole business is now becoming one integrated whole. It has to be looked at, and the importance of this Chow's article is that he says that the whole crystal engineering effort, starting from Schmidt and then myself and Peggy Etter and things like that, later on Zeverotko, he started from the chemistry and the crystallography side, but he says that now it can go into chemical engineering, the same principles, the same interactions used to obtain compounds with desired physical and chemical properties. So that part is still the same, but he says that the scope of this subject is really now very vast. That is what he is really talking about. Now, today we know that crystal engineering with co-crystals can improve drugs with poor physicochemical properties. Venu, can you hear me?
Yes, sir. Yeah.
Venu, yes. Okay. Now, with poor physicochemical properties. Now, crystal engineering and enhancement of properties, this is what we want to do. And we do this with the co-crystal technology by trying to fine-tune solubility, permeability, and to the extent that these two affect bioavailability. Then we have also physical and chemical stability. Co-crystals can do this also. Tabletability and mechanical properties. Now, this is quite a wide range of things, and we have worked on a couple of these areas, not all of them. But today I will have time only to speak about the permeability and solubility aspects, and to some extent bioavailability. So there are many others in the audience who have certainly worked on tabletability, mechanical properties, physical and chemical stability, and so on. I am not going to be able to have the time to look at all this thing today. So I will focus a little bit on this solubility permeability issue. Now, one of the early papers in this area was written by Professor Nangia, where he gives a mechanism for why a co-crystal can be more soluble. I mean, this spring and parachute model, this was not the first paper where these words spring and parachute were mentioned, but this paper gives a very clear description of the process. So here is the polymorph, stable polymorph of the drug. This is the co-crystal. The red Swiss cross is the co-former. The co-former dissolves, okay? And then you get an amorphous phase, essentially, is what the authors say. This amorphous phase in solution kind of thing. And this is the very important stage for us, because this is what is happening at this point. This is the end of the spring stage. And then the parachute stage is the slow step, the metastable thing, which gradually goes down. And this gradual coming down in the parachute stage is this. It goes back to the stable polymorph of the drug, or it goes to a metastable crystalline form. Actually, we don't care about these things too much. It is the time that this species is present in solution that is important, because this is when the thing has to be in solution and it has to be permeable also. So this is the most important part of this process. So this is one of the first attempts at trying to figure out why co-crystals can be more soluble than the parent drug. They can also be less soluble sometimes, but that it's also done deliberately. But by and large, people started looking at co-crystals to try to get the API more soluble. Now, another idea which is very important as far as the solubility permeability conundrum is concerned is the salt co-crystal continuum. So all of us know that when you have a co-crystal like here, NHN, you can have basically a proton transfer so that you now get a salt. So this side becomes plus and this side becomes minus. So just by moving a proton back and forth, you can go from basically what is a neutral species to what is an ionic species. And the extent of proton transfer in the solid state is not predictable. A continuum exists between the two extremes. This is also a very important paper which first heightened and identified this problem. Now, the salt co-crystal continuum, we looked at it ourselves, and there is a very nice delta pKa rule. So if the delta pKa between the acid and the base is more than 3 pKa units, then you can say that you will get a salt, this yellow region. And if it is less than 3 pKa units, you can say that you will get a co-crystal. Don't worry about these red things. These were just cases where we never got a co-crystal for whatever reason. So generally this delta pKa is equal to 3 is quite a good rule, and it will tell you just a rule of thumb when you will get a salt and when you will get a co-crystal. This is actually quite useful and it is used quite a lot. Now, once again, like this Everotco review of 2004, this paper by Istali has been cited many, many times. And all these people really, many of them, talk about this salt co-crystal continuum. It is actually very important. I mean, in the beginning, when the salt co-crystal conundrum was raised, people thought it had more to do with IP issues, saying that a co-crystal could be considered as a new composition of matter idea, but a salt need not be considered as such because people have been making salts of APIs all the time. So this is something that was being mentioned in the early days, but today I would feel, especially in the connection with permeability and solubility, the salt co-crystal matter now assumes more chemical significance. We had an interesting case ourselves where we had a crystal structure with multiple symmetry independent molecules. You can see this is isoniazid and this is para-amino salicylic acid. Both are drugs for tuberculosis and they form a drug-drug co-crystal. But you can see that this is not a center of inversion even though it looks like one. And the two OHN hydrogen bonds here and here are slightly different. This is more salt-like, this is completely co-crystal-like. So in the same crystal, you will have both co-crystal and salt. Now, this is just a kind of anecdotal observation for a single crystal structure. But you can imagine the fun in solution, especially in the presence of lipophilic and hydrophilic media of various types, that this particular equilibrium would be quite dynamic, and this will have its inevitable effects on both solubility and permeability. And this brings me to the next thing, of course, this is review material. All of us today of the drugs discovered process belong to the BCS class 2 category, high permeability and low solubility. This is because the nature of the drug design itself, the whole computational process, has a tendency to prefer high permeability or more lipophilic kinds of molecules. Many of them, in fact, Zavarotko says that even more than 60 to 70% is in this BCS class 2. So the idea is to get this BCS class 2 into the BCS class 1. This is where we all want to go. Now, and the idea is that can we do this with the co-crystal technology? This is the main point here. Now, please remember the early slides which I said, percentage drugs in R&D pipeline, that cost and the time and so on. All this is an additional scientific problem. The drugs that you're getting are not in class 1. You know, that is another really big problem. Now let us go to the next matter which I have referred to briefly, drug-drug co-crystals. Drug-drug co-crystal was an idea that was mentioned in the beginning, and it's something that has been pursued on and off by various people. And in the main context of getting a co-crystal more soluble and so on, making a drug-drug co-crystal is not of priority. Sometimes, especially in this case, both the drugs do roughly the same thing, same job. Both are antiretroviral compounds for HIV drugs. So they are actually marketed together and we actually got a co-crystal of these things. So people were trying to take two drugs which both treated the same disease and then try to make a drug-drug co-crystal. For what reason, I sometimes really don't know. I think people were just trying. They felt that these drug-drug co-crystals, you know, would be formed and they might have some interesting properties. It was just some kind of a blue sky research. But we wanted to go a little further. Now you see the main thing in this ionic salt versus more neutral lipophilic co-crystal is the effect that this has on properties. Now a salt is going to be generally more soluble in an aqueous medium. A co-crystal is generally going to be of less solubility than the corresponding salt. I mean, I'm talking about two species that differ in the proton transfer. Now, conversely, the permeability, which represents the ability of this drug to go through the membrane in the gut and go into the cell region where it can actually go towards the target, that is promoted by a more lipophilic character. So a more ionic character of the substance is going to improve solubility. A more lipophilic character is going to promote permeability. So you can see this solubility and permeability are working in cross purposes. In this context, I would like to take up some recent examples that we have studied ourselves in our group. The first is this drug-drug salt hydrate of norfloxacin and sulfathiazole. Now, this is an antibacterial, this is an antimicrobial, and there is enough literature in the medicinal chemistry literature which suggests that it is good to have combination therapies of antibacterials and antimicrobials. So there is some reason to try to get a co-crystal of this, but I am more interested in the properties which it shows. Now look at the solubility first. The solubility is on the left hand side. This is, norfloxacin is a BCS class 4 drug, and you see it's very poorly soluble at pH 7.4. It is much more soluble under acidic conditions because of its basic nature. Now similarly, this is sulfathiazole, but then now you look at the co-crystal. The co-crystal is really soluble. I mean these are things done at various pHs, 7, 4, 4, 1.2, and this is the co-solvent. We put two solvents together. So generally the trend is the same. The co-crystal is more soluble. Now this would tend to suggest that maybe on the basis of what I've just said, that the co-crystal is more ionic than this norfloxacin and sulfathiazole when taken together. But then look at the permeability. Now here this is norfloxacin, BCS class 4 I said, so even the permeability is very poor. And then this is sulfathiazole, which is BCS class 2, which is permeable. But then look at the co-crystal. The co-crystal is doing very well indeed. And so we have a case here of something where the co-crystal salt hydrate, I should call it, the crystal structure showed that it was a salt. The co-crystal salt hydrate was not only more soluble, but also more permeable. So this means that a simple model for solubility and permeability offsetting each other, the model is, the reality is not so simple. There is something more going on here. So we modified the earlier model here, which Professor Nangia had suggested, and then we called it a modification to the spring and parachute model. We said that yes, you get a co-crystal, which is a crystal structure here, but we said the co-former dissolves only partially. It doesn't dissolve completely as in the earlier simpler model. So there is an amorphous co-crystal in solution. This now becomes a key intermediate in the whole process. Now what this does afterwards, we don't much care. It can go to a metastable co-crystal. It can go to an amorphous API in solution, that is this stuff coming out, the sulfathiazole coming out. And then this part of the picture is the same as the old picture. Okay, so this is the new thing here, the amorphous co-crystal in solution. Now what do I mean by amorphous co-crystal in solution? Amorphous co-crystal in solution means that this particular thing, it retains some synthon memory. The synthons are here all right, the heterosynthons are here in the crystal structure, but it retains some memory of those synthons. Because if this were not the case, then you would not have a situation that we actually find in practice. When you take a physical mixture of norfloxacin and sulfathiazole, and then you take it through the membrane, we did the permeability test. The sulfathiazole goes right through, and believe me, I saw the experiment myself. You can see the norfloxacin crystal sitting on this side of the membrane. So it just separates the two just very, very nicely indeed. When you take the crystalline salt and do it, however, both norfloxacin and sulfathiazole go through just like that. So this is what I mean by synthon memory, extended synthon in solution. There is something here. Maybe it's helpful that we have started with a crystalline salt hydrate, so that there is some reason for the water to be held. And in this whole business of solubility, permeability, let me emphasize the unique nature of this water as a solvent. It can do all sorts of things. It's got a very high dielectric constant. It can make ionic things look less ionic because of this high dielectric constant. And I think it has an effect on permeability as well. Now, suppose this structure is sufficiently rigid and sufficiently stable for a short period of time, but long enough to go through the membrane, then you're through. Then you've got both the things coming in. So this is a sort of, what this paper told us was that unless you had some extended synthon in solution, you are not going to see the results that we saw. A co-crystal in vivo is different from a physical mixture of the two components. That's for sure. It's quite another matter. FDA says that at the very end near the target, only the drug should get to the target, but the target comes very much later from these steps. This is what is happening in the intestine, the membranes of the intestine, when we want the stuff to go through, and sulfathiazole is essentially able to drag norfloxacin through the membrane. So this was the first paper. Now the second paper, well, it came in the newspaper and so on. And can you see this? Or
Yes, sir. Yes,
you
sir.
can see
Visible.
it came in the Hindu. Do you see? It says presenting something. Give control. What is this? Give control.
From our side, it is all right, sir.
Okay, it's okay. So it came in the newspaper, and this is the man, Dr. Prasad, who I saw has logged in to the talk a few minutes ago. He is in Reddy Labs now, and he did most of this work. And we even got an Indian patent for this recently. We had to wait a long time before we got it, but antibacterial, antimicrobial composition and method of preparation thereof. So this was the first step. Now we go to the second paper. This is mostly the work of Palash, who is now in SRM University in Chennai, and Dr. Somnath Ganguly, who was a postdoc in the group. Now, hydrochlorothiazide is again a class 4 drug, which is very, very poorly soluble. And in this study, we made co-crystals of hydrochlorothiazide with a number of co-formers, no drug-drug here, it is just with co-formers. And we looked again at solubility and diffusion slash permeability enhancements. Now, what did we find? Now, this is HCT. These were the co-formers, which are all pretty standard compounds. And then this is the solubility and permeability behavior. Now let's take solubility first. The black graph. This is HCT, which is poorly soluble, as I said. And then these are the co-crystals, which are all more soluble, except this one with the succinic acid diamide, which is an outlier, which I'm not going to discuss too much. But you see, most of these things, the solubility is doing quite good. And when you come to permeability, you can see solubility and permeability are roughly inversely correlated, as they should be. So the simple model for the solubility and permeability being inversely correlated is not such a bad model. So, but when you take the product of these two, if it was perfectly correlated inversely, the product of solubility and permeability should be a constant. It is not a constant. For all the co-crystals, it is except the succinic diamide, the product of solubility and permeability is more than that of pure HCT. Now, this is again telling you something, that solubility and or permeability are controlled by factors other than strictly the ionic character versus the lipophilic character. Among the two, I have no way of distinguishing these things now, but among the two, my feeling is that it is much more difficult to predict solubility rather than to predict permeability. I know people will disagree with me when I say this, but I think it's a much trickier matter to figure out whether a co-crystal, what the solubility of that co-crystal is going to be and its rationalization, compared to the permeability. But whatever it is, the important thing in Palash's paper is that the product of solubility and permeability is always better in the co-crystal. Now, this is something which could be a very general observation for co-crystals. Co-crystal is just not a new drug or a new thing like a polymorph or something. You know, polymorphs sometimes change the bioavailability because they're very insoluble and so on. But here is a more general thing. Will most co-crystals improve the solubility and the permeability of an API? I mean, this is the question which I ask you. This is all open things, and you know, it's not my idea to give a talk where I have all the answers and all the questions. No, but I think these are things that people need to think about. Are co-crystals going to be then very, very important? Because simply by the very nature of forming a co-crystal, both solubility and permeability go up. Is the behavior of HCT specific for HCT, or is it something that is going to be important for more co-crystals? And this brings me to the third paper of ours that I wanted to discuss, which took off from a paper published by Dr. Hutchins on trimethoprim, which is an anti-urinary medication. And this is again, I believe, BCS class 2, which is not very soluble at all. So they wanted to make this thing more soluble. And so they made a lot of co-crystals, bipyridines and so on, yields neutral co-crystals, etc., etc. And they were trying to improve the solubility of this. So this is the paper that got us thinking about trimethoprim in the beginning. And what we then did was, we looked at a co-crystal that we made between trimethoprim and another anti-urinary medicine called nitrofurantoin. Now this nitrofurantoin, there are no clinical issues with this material. Technically, it is supposed to be class 2 or something, but the oral bioavailability is good and much of it is even excreted. So it doesn't look like there are many problems with nitrofurantoin. Why did we try to make this co-crystal? What we saw in this particular co-crystal, let me tell you what we obtained. These are the two things, and what we made were a co-crystal hydrate, and then we made co-crystal solvates with a number of solvents, organic solvents, acetone, dioxane, THF, DMF, aniline, and so on. And these crystal structures differ slightly. We also got an anhydrous form in the very end, where we couldn't get a crystal structure. We just resisted our attempts to do that. But if you take the first one, nitrofurantoin trimethoprim hydrate, the ionic hydrate, it has this imide aminopyrimidinium synthon. This is the imide, this is the aminopyrimidine. Now, in the aniline solvate, we found that you take this imide and you flip it around, and then you again get another imide aminopyrimidinium synthon, but this is an isomer of this. And in all the other solvates like acetone, THF, etc., the other organic solvents, we got the co-crystal form. So once again, the salt co-crystal continuum plus this kind of thing. So as you can see, electronically, this is a very labile system. There are charges moving all over the place. Sometimes, and I mean, what is the meaning of this different crystal structure, this different crystal structure, so on? It means that under a suitable in vivo condition, you can have all these kinds of situations prevalent. That's all. The co-crystal gives you a kind of a snapshot of what is happening in solution. So if you get more co-crystals, you will be able to analyze more environments in solution. And as once Professor Sundarlingam told me very long ago, almost 30 years ago, he said that we were talking about protein crystal structures, and then he says, you know, Gautam, he says, the protein structure, even with a lot of water in it, may resemble solids more than it resembles solution. And when I looked at this example, then that old thought came back to me. And so I felt that we must start looking at these things as a rather dynamic interplay of charges and polarity. And in this kind of a situation, these are other synthons that are also available here. We can try to understand. What we did find is that the bioavailability of this nitrofurantoin trimethoprim ionic hydrate, the bioavailability was better than the individual drugs. That we found. So which means both solubility and permeability, or both, they are all going up in this particular thing. Now then the question arises, what is the significance of all these things? And we have written this in our recent paper in, well, this is how the synthon actually looks like. This is an interesting picture actually. And this was produced by one of my postdocs just last week. So as you can see, even with this COVID and other things, and with me largely isolated at home for 14 months, still something is going on. I won't say much is going on, but something is going on. So you can see here, this is the synthon, this is the aminopyrimidine, and this is the imide. So you can see this is the hydrogen bonding part, and then here is a CHO, believe it or not, between the methoxy group and this one, and here's another CHO. I was very pleased to see this one here. This is the nitro group, a nitro to CHO. This is nitro is a very good CHO acceptor. So this thing may be robust to some extent, and look at the space filling model with all the hydrogen bonding groups pretty much taking care of each other. You know, this looks like, to me, it looks like a lump of oil. And you can start understanding why the cocrystal will definitely be more permeable. Now, why it should be more soluble, I think is a far more difficult question to answer. But it is also certainly more soluble. There is no question about it. The experimental results show that. So once again, you come to this same conclusion that we saw in norfloxacin sulfathiazole, that when you get a cocrystal, somehow you are improving both solubility and permeability in some sort of a general way. This doesn't seem to be something particular to these compounds. And I think this is an aspect of the subject that needs more looking at now. And so then I take you back to Chow, you know. He says crystal engineering addresses crystal structure and its fabrication, he says. And where is that mouse? Okay, fabrication, pioneered by Schmidt, etc., etc. It is a highly interdisciplinary field that involves various and often uncorrelated areas, like this diverse. See, today's uncorrelated is tomorrow's correlated. Uncorrelated is simply our way of expressing that we don't understand something. From an industrial perspective, now he comes to industry straight away, encompasses broadly the application of basic principles of crystal chemistry and materials to all this tailor-made thing, followed by formulation development. People have started writing about formulation development. Venu had a nice review on this last year, and Mike Zavarotko had a good study on formulation where he talks about, I think, the pH of the surface, you know, and then he has a new quadrant of class 1, class 2, class 3, class 4, this time not in Roman numerals but in Arabic numerals. I should call them Hindu numerals now, that's what they say. And where he talks about the acidity, surface acidity of these particles and process design. So solid state knowledge covering important material properties, polymorphism, salts, amorphous, blah, blah, manufacturing, processes, coming very far from, you know, topochemistry and cinnamic acid. Manufacturing processes, appropriate crystallization, designed to produce a pharmaceutical product. Now, and he calls it nicely, throw off the wall. He says, in the earlier days, the chemists, the process chemists and the computational chemists would simply give a molecule and then throw it to the others and say, okay, you guys do whatever you like. Make a cocrystal if you want, make a polymorph. Any problem that you have with various issues like solubility and permeability, you please solve it with formulation. Okay, you can solve many of these things with formulation, but after some time, formulation becomes expensive, very cumbersome, and it reminds me in a way, in the old days when people did synthetic chemistry, they used protection, deprotection methodology. It was clumsy, but it worked. But then later on, ecological and other concerns said that this is too expensive a way of doing it. I would like to suggest that the present kinds of formulation strategies are the supramolecular equivalent of those old protection, deprotection methodologies. Why don't you think in terms of changing the molecule itself earlier in the process, instead of getting something which you then claim is perfect and then throwing it off the wall? The essence of the argument in our paper is that crystal engineers need not necessarily hand over a clinically perfect co-crystal. Co-crystal is not something that follows after the active molecule. There is no need to have the sequence between chemist, computational man, crystal engineer, then the industrial chemist, pilot plant, final plant. There is no need to have this. There should be a lot more of give and take, talk between these various sections. And this is where I would still like to go one step further. And this is something we hinted at in this paper, and which is what we are trying to work on. Now, if I go back to the synthon, if I go back to these synthons, the suggestion I'd like to make, again for your consideration, is that the molecular features, including the hydrogen bonding groups and so on, are important as far as they are target directed. So finally, that is the medicinal part of the thing. They have to get to the active site and then they do something, a complex series of biochemical transformations take place. But what if, and if you look at the results of the 3 papers I have mentioned on norfloxacin sulfathiazole, or the hydrochlorothiazide, or the trimethoprim nitrofurantoin. What if it is the synthon that encodes the combination of solubility and permeability that is going to be most important at the stage of get the bioavailability? So it may be that the molecular synthons are doing the medicinal activity, but it is the supramolecular synthons that are actually responsible for this balancing of this solubility, permeability. So if this is the case, and this is something that is right now even pre-hypothesis, if this is the case, then one can go beyond Chow and we can say that we can also use the synthon, and this is important, we can use the synthon as a supramolecular pharmacophore. Pharmacophore is known because they take the medicine and then they put it on the active site and then they keep looking at that and try to find other molecules which will fit the pharmacophore. Define now a supramolecular pharmacophore, which will then lead you to properties. Now, if you take just an example, let's say using the trimethoprim example, there are about 15 molecules of marketed drugs which have the aminopyrimidine skeleton. There are about 5 to 6 molecules that have the imide functionality in a marketed drug as in trimethoprim. So if you just were to take a combination of these synthons, roughly there will be 100 or so drug-drug co-crystals that involves this aminopyrimidine and the imide. 100. You can make them in the lab. That's not difficult. Some of them will work, some of them will not work. When you get a co-crystal, if you were to check out whether the solubility and the permeability are improving in this case, then getting good solubilities and good permeabilities need not be a matter of chance. You can then start looking for it. And I would like to suggest, therefore, at the end of the talk, that you start looking at this whole co-crystals, not even as, but beyond co-crystals and industry, beyond chow. These co-crystals can actually be used to get molecules that will help you greatly in this stage. Because you are targeting properties which otherwise in the normal drug cycle would come up much, much, much later. And as they always say, it is better to lose cheap. And looking for a supramolecular pharmacophore is as easy computationally as looking for a molecular pharmacophore. So this is, I think, an idea that we should start thinking about, because otherwise there is no reason why these solubilities and permeabilities should go up together. At least to me, it doesn't appear very clear right now. More experiments certainly have to be done. And then, of course, these are all the success stories. These are all medicines that co-crystals that have now been approved and they are being marketed. We mentioned them in our essay in Engagement 2 years ago, and Mike Zavarotko and others have given a detailed description of these things and some of the background involving this. I mean, so this is all just the beginning. I have no doubt in my mind that unlike polymorphs, which were kind of a speculative, black magic sort of thing, co-crystals, I think, represent a real good chance for crystal engineers of the chemistry and the chemical engineering type to get together and try to develop a much better, long-term, cheaper way, more sustainable way of handling these drugs, not the present expensive way. And I think this is something where a lot of us need to get involved in. This is a multi-skill thing. We have to have big collaborations. It's not possible for one person to sit in one lab and come up with something that will change the world. But I think this is something for both academics and industry people to look at and to think about, extremely carefully. And finally, in conclusion, I don't know, I cannot see the time here, but I think, oh yes. This is formulation. I mean, the first thing was it is becoming important in formulation, and then solubility, permeability are becoming important. They have to be considered together, and they are affected by very complex factors. This is not simple. It's not just a matter of ionic and lipophilic. And then the heterosynthon encodes within it the synergistic property of the co-crystal. So synthon itself, when I suggested it in 95, I said it was not just structure. It was not just about structure, but it had to do at all stages of the crystallization game. And the fact that, the essence of the supramolecular principle is that there is more to the synthon than just the things that make up the synthon. And so it may be therefore used much earlier in the drug design, and it will be very attractive if we can target the medicinal activities through the molecular synthon and the bioavailability matters through the supramolecular synthon. This would then, bring the whole thing in a different kind of a light. And therefore, this is the open question which I leave here. Can we therefore go towards a heterosynthon as a supramolecular pharmacophore? This is an open question which I leave to all of you. And again, in this COVID time, one has the luxury of, talking a little bit, thinking a little bit more, reading a lot more, not jumping around so much as we used to do. And I think it's also time for you people to take these suggestions. This is not a published work, this last thing, but it's an idea which I'm giving to you all to see if there is anything that can be done, because today's databases ways of collecting data, artificial intelligence will make all this very, very easy. So we can get any number of co-crystals on paper and then try to work it out and see whether you are able to get the desired properties, whatever things that you kind of want. So this sort of thing, Venu, this is about 1 hour, so I don't know how much time there will be for questions because I didn't have a clock over here on this computer, so I couldn't see what the time was. But I think it's about an hour, and so I think I would like to, with this at this point of time, thank you all very much for your kind attention.
Thank you, sir. Thank you very much for this great, fantastic talk. Can I request Professor Nangia to unmute himself and ask? Professor Nangia has some question.
Yeah, thank you, Anand. Am I audible?
Yes, yes.
Yeah, thanks. If we have time, I had posted it in the chat box, and my query was about the close-packed cyclic synthon structure which you had shown, and you said it could be ascribed to the higher permeability. And this is more of a query comment that it would mean that such strong and weak hydrogen bonded multi-molecular aggregates are surviving in terms of time and also in solution long enough to reach the semi-permeable barrier and then exert their effect to be seen as a visible property.
I'm the wrong person to ask about weak hydrogen bonds. So, you know, given these the kinds of non-aqueous kinds of conditions in the membrane and so on, the CHO may actually be better than the strong hydrogen bond under those conditions.
Okay.
I have, sir, one quick comment after going through your the first. I have read your papers and especially the definition paper in crystal growth design several times and referred. For our patent, from each and every country, from China, USA, as well as now in India, the major point which we have to highlight every time and send your papers in argument, people don't still understand differences or don't get attorneys especially get confused on the definition of co-crystal. So it is really a most important thing to make it clear, and that is it is very useful for us, and especially for a person like me who is not an organic chemist or crystal engineer basically.
Well, they will listen to the FDA 2018 guidelines.
Yeah.
If they don't, even if they don't listen to any of us, so they listen to that. And you know, the European definition is a lot closer to the recommendations of the Delhi meeting.
Yes,
it's a lot closer, and it's a very helpful definition too. So I think yes, I mean any subject goes through this, Anand, because I always like to say that this whole business of co-crystals and pharma co-crystals is one where the academic and the industrial things are almost going in parallel. There is not much of a time gap between these two, which is what makes it interesting for both groups of practitioners.
And especially I'm very thankful on behalf of CPS. CPS is coming out with first product at 500 kilograms. Product will be on the market maybe next month or so.
Very good.
Rafael,
Professor Xavier, Dr. Xavier Elana, can you unmute yourself?
I can't see him.
Yes, thanks. You can hear me.
Yeah, I can hear you. That's important.
Okay. Thanks a lot for your talk. It's really a pleasure always hear the talk of Professor Desiraju as a great mind. I love the idea of the heterosyntons be the new pharmacopher. It means that that's the key idea of the talk. And I wanted to ask that if it's really something behind that idea, we will seriously have to revisit all the work that has been done for the area in the last, let's say, 15 years, and trying to look everything with the new eyes. And this will mean a very big change of game in the last year. What are your thoughts about this could change?
Well, Javier, let me put it this way. Science is inherently iconoclastic. And so, we do something today and then tomorrow somebody comes and, the whole thing has to be revisited. But I don't think it's a complete reversal of things. I think it's looking at some of the older examples. And one also wonders, I was always mystified and of course pleasantly surprised that so many of these co-crystals had better solubilities and permeability. This is something that I generally noticed, not just from our papers, but from papers written by so many people. And I don't think this is a case where people are only putting their success stories forward. I think it seemed to me that it's a fairly general phenomenon. Firstly, it's very easy to get a co-crystal of an API in general. There are a few we know molecules, drugs, aspirin, for example, doesn't give many co-crystals, considering the amount of effort that has been put into that molecule. But many of these drugs seem to give lots of co-crystals. So I think one should look at it a little bit more. And I would say, even in terms of this revisiting, if something can be done computationally, I think today our advantage is, superior computing powers, this cloud, this artificial intelligence, huge databases, not just the CSD and so on, but the medicinal databases themselves have got, we need property databases for sure. I think this is something that crystal engineering field is missing badly. And it should not be just a structured database. And I was telling someone a few days ago, I want to take this whole idea, at least whatever is in my head, take this crystal engineering idea from structure definitely into properties and processes. I think that is the way it should go. And everything in science is based on what has been done before. I would feel that the progress here is pretty fast. 2004 Zavarotko, 2007 Chow. Then, by 2011, the first FDA thing. By 2018, already a much better definition. And the number of papers that are being written in so many journals. It's not just CrysTechCom or Crystal Growth and Design. It is Drug Discovery Today, it's Small Farm, it's so many places. And I think lots of people are thinking about it. And about the lawyers, I think, Anand said and others said, I think that the lawyers, they finally listen to what, in the end, what the scientists say. The lawyers cannot make science. Okay, so, they don't even try to. They don't even try to. I mean, let's be fair. And
I know that.
no, they don't.
We have to
And
face
so,
the same.
if it's a thing that, as I said, when we are not able to define something properly, I've been involved in many of these nomenclature issues. Whenever people, and fight about nomenclature, it means the understanding is incomplete. That's all. Yeah. Otherwise, people don't get, scientists don't get emotional,
Yeah.
We should not get emotional. And, there's a name business. Useless, junk. So it means you don't understand something and then you resort to emotion to try to solve the problem for the day. So I think it's okay.
Professor Steed has come, has
Who
some
is this?
idea on the Professor John Steed.
Oh, hi, John.
Would you please unmute and ask the question that you brought up with the interester?
Hi, Gautam. Lovely talk. Thanks a lot. I wasn't really asking a question. I was just being a little bit provocative in the chat and saying that Entresto is a double salt hydrate, not a co-crystal. But everybody
Yeah.
yeah.
For
the younger people, I responded.
In fact, I think this is another thing. The interesting point that you bring. The more complicated the classification becomes, the poorer is your understanding of the phenomenon.
Yeah, that's very true.
So it's a question of frame of reference. Now I've shown you that trimethoprim, a nitrofurantoin example. Based on the polarity of the medium, it can change the synthon and the charge and so on. So how does it matter that, where the charge is and so on and so on. But the other thing I must tell you, there was a paper, I think Christer wrote it long ago. I didn't cite it today. He says, Salt-cocrystal, does it really matter? I think it really matters. It really matters today because of this business of solubility and permeability. and the fact that they are not perfectly inversely correlated. And that lack of that perfect correlation means that there are other factors going on, and I don't know what it is. It could be my hunch, but I think solubility is going to be much, much harder to understand than permeability. This is my feeling. I could be wrong. So that's a nice thing about these online talks. You can say what you like.
Dr. Rafael Prohens.
Hello, Rafael.
Hello. Hello, can you hear me?
Oh, yes.
Yes.
Hello, thank you very much for this nice talk. And I have a comment about bioavailability of cocrystals, because as you mentioned before, it's very easy to impact on solubility of an API by using a coformer which is much more soluble in water, for instance. And as you also said, it's very easy to synthesize new cocrystals. It's a matter of time. But I think it's very hard to impact on the real absorption of that particular compound in vivo.
Correct.
And I have a very frustrating story during the last years trying to improve bioavailability of very hydrophobic compounds.
Excuse me, improving the bioavailability of what?
Very hydrophobic compounds.
Hydrophobic compounds, yeah.
And as you know, in the pharmaceutical industry, every time APIs are more hydrophobic.
Correct.
And I think that it's what really matters at the end is to find a good formulation that takes advantage of the increased maybe solubility of the cocrystal. But if you don't have a good formulation, a good way to help the compound to enter into the cell through the gastrointestinal tract, there is no way to do crystal engineering. And I think that most of crystal engineers are crystallographers, they are chemists, but I don't have much experience designing a good formulation. And in many hydrophobic compounds, it's, most of them are absorbed by endocytosis. So it's very difficult to translate the solubility, the increase of solubility into the higher levels in blood. So what are your feelings regarding?
Okay, see all the points you make, Rafael, are very, very valid, and it is correct. I am not proposing here that we junk formulation altogether. I am just saying that when in the present system, when the chemist, let's say the computational person, then the chemist, and then the scale-up person, they're all disconnected from each other. And so that finally when a molecule is given, it is taken as final. And already too much time, effort and money have been spent in getting that molecule and the clinical trials and so on. So nobody is prepared to change at that point. And therefore you have to put all your eggs in the formulation basket, which is very important, no doubt. But there comes a point, India is supposed to be the formulation capital of the world or whatever it is, so many slogans we hear. And we get into formulation because we can't do other things, maybe, I don't know. But you hear a lot of hype today, too much hype after COVID, all sorts of things. And you talk about lawyers, imagine scientists talking to politicians today. Every day there's a new theory in the TV. Now, so when you come to your question, all I'm saying is that if there are ways of losing cheap very early in the design aspect itself, so that even if you had a hint that this molecule will be slightly better for solubility, permeability compared to another one which looks chemically somewhat similar, then you should have enough confidence to tell the synthetic chemist, I don't care if that benzene ring doesn't have a methoxy group, you better go and make it. Today we don't have that authority. He or she comes and tells us, this is what we can make, so this is what you take. So I think the crystal engineer, by that I mean both chemist and chemical engineer and the process person, the person who's going to have to do these terrible formulation problems and solve them. All these people should have an input into the very earliest stages of the design game. And in that case, you are just spreading your losses throughout the process instead of starting with some fixed point and then saying, you guys do whatever you like, but we don't care. This is the molecule. That's all that I meant. You can't do without formulation. But I'm saying that today it seems in some of the cases, the formulation has become, the tail is wagging the dog now.
Right. But crystal engineers are not formulators, and formulators are not crystal engineers.
But this is what Chow says. He says Chow and then Zavarotko and then Venu the other day. Their recent reviews on crystal engineering and formulation tell you that formulation is very much coming into this crystal engineering umbrella. It is. It was not the case before. Past tense, you are correct. But I think in the present and future tense, this is going to change. And this is what, Javier just said, we have to rethink, this rethinking has to be done in these kinds of domains.
I agree. Thank you.
Prof. Nangia, do you have a question you raised? It seems, yeah.
Yeah, if there is time, I think my first question was lost halfway. The point I was trying to ask Gautam, and there are others as well, and perhaps if the thinking is wrong, maybe just correct me. My understanding is permeability is about the hydrophobic character of the drug, as you rightly showed with the closed packing model. But in the end, the hydrophobicity of the drug has to do with the binding of the drug to whatever is the final receptor of the drug, so that it can exert its therapeutic action. And so the point I was trying to get across is, while the first part of the hydrophobicity requires the synthon, in the end, the drug has to dissociate, and only then, as the individual drug, will it be able to bind to its target and do its drug action.
This is a fair point. It's a fair point.
That is what I was trying to get
at. The hydrophobic portion also has a molecular function. It has a molecular function, especially in kinases and all, if you see. Although it is the hydrogen bonding groups that bind directly to the active site, the rest of the thing also has to match properly, no doubt.
I have a question, but I will let the members from the Brazil, North America, and South America to give some priority if they have any. But Len, are you there?
Professor Price.
No, I saw Len very briefly on the thing. I don't know if he's still there or.
Yeah, sir, I have more than a question. It is about yeah, Len, please go ahead.
I'm fine. I just enjoyed your talk, Gautam, very much.
Oh,
thank you.
Thank you. Recently, we have published a cocrystal permeability paper with Palash. Palash is also one of the audience today. We had hard time justifying them relationship between the crystal structure to the permeability property. How far are we now? You touched upon already what could happen with the synthon. Further, I mean, how to go about this?
There are so many journal editors here in the audience. I think you should ask them this question. I see John and I see Len. I see Peter. I don't know. I mean, it's a question. If it's your idea and you want to fight for it, you have to. I mean, that's the way in science. And it should be done in a reasonable way that passes the tests, in today's state of knowledge. I mean, that's it.
Are there any further questions?
Can I ask a question? It's a bit general. I would like to ask Gautam's personal opinion. Why does he think that we don't have so many marketed products based on cocrystals?
Why don't we have?
Why do you think we don't have many marketed products based on cocrystals? Because there is a lot of science behind that. There are several publications. We almost know everything. but I will agree with Rafael. There are issues regarding cocrystals and formulation. But what is your personal opinion? Why do you think we don't have it? Is it
I don't know. I'm not disappointed, Dennis. I'm not disappointed with the 5 that I've already put up and which are well known now. And I think people are going to go to co-crystals only when the existing single molecule drugs don't work. There's got to be a special problem with the single molecule thing before people start looking at co-crystals. After another 5 years, 10 years, then people will go to co-crystals as a matter of course, because they will enter the regular domain of compounds. Okay, so right now we're still early days, but I'm not disappointed. I think 2014, 16, 17 years, I think we've gone quite a long way in a very conservative industry, mind you.
Yes.
This is not something that changes very quickly or very easily.
I just have one comment from my experience, because we work with some local companies. One of the major issues that they're really afraid of that is the stability of co-crystals. When you start putting them under ICH stability settings, then you might find they might dissociate co-crystals. And this is what it goes back to Rafael's comments, that you have to design a formulation that protects the co-crystal.
I don't know if I forget the reference, Dennis, but Mike has got a paper on this recently. I mean, where in the formulation process, the co-crystal was dissociating. That's when he defined this, the class 1, class 2, class 3, class 4, based on the pH of the surface particles and so on. So I forget the citation, but I think he has actually described a case like that, that the decomposition of the co-crystal during the formulation is a real issue. It's not, it could be very general also. I quite agree with you.
Thank you.
Professor Price, do you have any comment from computational point of view?
I think there are 2 things. The computational side tends to go for the thermodynamic solubility, which is can be very different from the measured solubility, which is kinetic, which is difficult. And I think there are great problems in actually defining what solubility you need for the sort of databases or property relationships, because of as Gautam has very successfully shown, the complexity of the processes that are going on, particularly for co-crystals, but possibly also for even single component systems. And
excuse me, Sally, I have a question on your comment. Do you think that some kind of molecular dynamics approach, things like that, would be better for the kinetic solubility?
Yes, but with the computational side, we are running into the problems in so many spheres that the accuracy that we would like to get for crystal energies as a function of temperature require methods that are not suitable for molecular dynamics. We don't have, the whole computational modeling has a mismatch between the methods that are relying on molecular dynamics versus the static methods, just because the force field development, particularly for drug molecules, often isn't good enough, because the functional groups are different. And also, for so many drug molecules are now so complex that actually the van der Waals contribution can be rather dominating over any of the hydrogen bonding contribution.
Okay, thank you.
Yeah, Professor Ayala.
Okay, thank you, Professor Desiraju, for the nice talk. I found very interesting the idea of from your comment and comment from Professor Nancy about the keeping the synthon as a unit to improve the permeability. But I was wondering about that, because what all the regulation agency agree is you must have completely this dissociation of the cocrystal to consider the cocrystal as not consider the cocrystal as a new molecule, especially in the case of the repurposing of drugs. But if you consider that you keep the synthons during the permeability, it will not be an issue regarding to regulations?
I don't believe so, because I think what they are talking about, I mentioned this in the talk myself, that it happens at the stage when it gets to the target. And I think Professor Nangia has shown this for temozolomide or something in that somewhere. That I think the going through the membrane in the gut is still at an earlier stage than the stage at which the FDA is concerned about it in its 2018 guidelines. So I don't think that will be an issue.
Thanks a lot.
Yeah, just to add to that, I think the synthon should carry up to the N-1 stage, but not the nth stage, because if it goes to the nth stage, then it will be counterproductive.
Yeah.
Definitely, because it's also eating up the hydrogen bonding groups, which are important for the recognition.
It's like a rocket launch where you have to sacrifice your lower module at some point so you can get the next acceleration.
Okay. Len has. Hi, Len.
Yeah.
Unmute.
Gautam, yeah. So if I was understanding the message near the end correctly, if you were to start a drug company from scratch today, okay, would there be a preference to have your co-crystal group at the back end or a little bit closer to the front in terms of beginning to study drugs that exhibit maybe promising bioactivity, and perhaps the co-crystal approach could allow you to really improve things right away versus having to do extra work in terms of the bioactivity?
How many people would you have in the company?
Well, that's a good question. Actually, you might be able to have, might get
Because
more productivity.
I think I would start in the short run. I would keep some of the co-crystal people closer to the front end, with an option that in say 3 to 5 years, I will start pushing them more towards the back end.
Right.
And if I have at least, let us say 5-6 people in the co-crystal domain, that would give me enough room to juggle these two groups.
I agree. I think earlier is probably a direction to go.
Yeah.
Okay.
Yeah, it's like kinetic and thermodynamic. I mean, we have to solve the problem. I mean, tomorrow with this pandemic, we have to give the vaccines and get the people, cured, and then later on we can worry about medicines for the COVID patients, once they get hospitalized and things like that. So,
all right.
Just a quick comment to Len's comment. I heard a very senior medicinal chemist say recently that in all the databases which exist either in companies or in public domain, he said we have enough active drug molecules sitting in the world. The question is which ones to pick them. And so I think if we could reverse the priority, you could pick the good ones out of that ocean of a database using a different approach.
Yeah, but that's like saying all knowledge exists in the library. All knowledge exists in the library, but you don't know which book to go and pick up and read. So
yeah,
it's truthful, but not very useful. I'm afraid.
Yeah. Okay.
Yeah. Professor Anand. Yeah.
Yeah. Thank you very much, Professor Desiraju, and all the participants. We have excellent. I don't think we had such an opportunity to get all stalwarts in one go. Thank you very much, and I'm looking forward to hearing.
Thank you very much, all of you.
Thank you
In your various time
Gautam,
zones,
and
being
everyone.
able to take the time out to come and listen to me, as these are very, very anxious times.
Thank you.
Hopefully we will all get through this thing in one way or the other, and in any new normal that emerges, I trust that we shall all be in a position to even see each other again at some suitable opportunity. In the meanwhile, everybody stay well and stay safe above all.
Thank you. Thanks a lot.
Thank
Bye-bye.
you, sir.
Bye, everyone.
As usual, thank you very much for your visionary thoughts.
Thank you, Benu. Thank you, Anand.
Thank you very much.
Thanks.
Well.
Thanks, Venice. Thanks.
Thank you all.
Bye.
Thanks all.